Welcome to this case study. We're going to explore here a case for an RNA-Seq workflow that is designed for the discovery of novel cancer drug target genes. We're going to discuss how to manage the laboratory data that are produced in this process, but also how to integrate an automated NGS data analysis pipeline into a single platform. We're going to make use of a couple of neat AWS functions like AWS batch or step functions that help you to scale your bioinformatic pipelines and enable you to analyze tens or hundreds of samples in parallel. My name is Jonathan. I'm the founder of EVOBYTE Digital Biology. If you have questions or comments, please leave them here, and we're going to start now. We start by looking at the RNA-Seq workflow that is part of the research process and that is designed to compare the gene expression, so the abundance of individual genes in a tumor tissue compared to a matching healthy tissue. And for this, we start by isolating RNA from either of the tissues. The RNA is then processed into next generation sequencing libraries that typically involves ligating adapters and performing a PCR enrichment. The libraries are then typically pooled and sequenced on an Illumina machine, which leaves us with our raw data, which in this case are FASTQ files for each of the samples and libraries that we sequenced. And then it's part of the bioinformatics pipeline to process these FASTQ files and perform a differential gene expression analysis, DGE, in order to identify genes which are upregulated or downregulated in a tumor compared to the healthy tissue. And those make up then the potentially interesting targets that we can explore further. First, we're going to define a data structure for our research process, and this data structure should reflect the raw data and samples and define their relationship. And this data structure we are then going to translate into a relational database scheme and a user interface for people to actually interact with the data produced in our platform. And in this case, we will define a project, and each project can have a number of samples. Both have a name, a description, and in both cases, we want to be able to upload some files that are related to the projects or to the samples. And importantly, for each of the samples, we want to be able to store certain features like disease type, replicate information, tissue information, everything that helps us to later on guide the analysis of the data sets. On the other hand, we have raw data. Those come from our sequencers. As we said, it's mostly FASTQ files, and these FASTQ files we want to link with the samples that are stored in our database in order to be able to reference the features and metadata throughout our analysis pipeline. So whatever data analysis we put on top of the raw data, we can always go back and via the raw data get the information for the samples and through these metadata structure our analysis. So let's check how to translate the data structure that we discussed before into an actual backend application. We make use of Django here. Django is a Python web framework that is compatible with different databases. In this case, we make use of the PostgreSQL database, and we use Django in combination with the Django REST framework in order to provide a RESTful API for interacting with the backend later. We start by making use of the Django user management that allows us to create different users and manage permissions of each of these users for different views of the database. And first we define a set of models that come from the structures that we defined before. So we make a model for the project, for instance, and derive from this we have a couple of models for project files, because we wanted to be able to upload a couple of binary files, which could be Excel files or images or PowerPoint presentations for each of the projects. Then each of the projects can have one or many FASTQ samples. Those are actually the libraries that are processed within our RNA-Seq workflow. As you can see here, there are a number of tables attached to each of the FASTQ samples. First, a FASTQ QC table. We will have a look at the bioinformatics in the second part of this video, but this table is meant to store QC information for each of the FASTQ files. We have an annotation table that holds the metadata or features for each of the FASTQ samples. Those were these categorical variables, for instance, the age or the tissue type or the replicate information that would drive our analysis. And we keep this here in a separate table because it allows us to basically version the annotations that we define for each of the FASTQ files. And finally, also here, a file model that allows us to upload a number of files for each of the FASTQ samples. So for instance, some metrics that come from the NGS library preparation. Then each of the FASTQ samples can have one or several RNA-Seq datasets attached. And those columns basically, or those tables, hold the information and data that come from the RNA-Seq data processing. We will also talk about this in the second part, but basically the fact that we allow here several of these datasets attached is related to the fact that we want to be able to also version the RNA-Seq pipelines, and if we update this, we can store different versions of RNA-Seq data for each of the input raw data. And finally, we have this DGE results table. This is basically storing the results from calling the differential gene expression analysis, which we will also see in the bioinformatics part. Then we also implement a number of AWS service calls in our backend behind a REST API endpoint. This allows us to trigger specific tasks that are running on AWS. For instance, in our case, we implement the RNA-Seq data processing as an AWS batch job. And this allows us basically to trigger these tasks using a URL. And this also allows us to use the same user management that we use for accessing individual views, also for accessing certain services by AWS. And this is why we implement this here in the backend. And we will have a look now at the user interface and how we can populate these individual tables. So this web application here is written in Streamlit. For those who don't know Streamlit, it's a Python library to develop web apps and interactive dashboards. It's great to use for plotting or visualizing data. I'm going to link it also in the notes. And we start now here at the projects page. So the projects page is basically our hub to manage different and organize FASTQ files into different projects. We have now already created a project P1, which is the project that we are going to attach our sample data to here. And we also have a default project, which is always created. And new files which are uploaded into the data platform are actually assigned to this default project. So what we wanted to implement here is also the option to upload files and assign them to individual projects. And this we can do here in this metadata page. Here we can just click on upload a file to project. We're going to select our new project. And what we're going to upload here is actually a budget file. So let's say we want to systematically record the budgets for all the projects that we are doing over a year or so. Then we can attach the file here. We can give it a name and we can upload this and then also store it in our database linked to the project. And that's really an advantage because let's say we want to do some systematic review or run some machine learning on our budget or some other types of analytics, then we have basically everything in the database attached to the project. So that saves us lots and lots of time going through some files or digging in some archives. With this approach, we have basically everything in this table. And next we come to the core actually of this platform, and this is of course the FASTQ dataset. So this is the raw data that come from the sequencer that are QC'd automatically and then loaded into this data platform. And we can see here we have a set of example datasets here from a tumor and healthy, and those are basically now assigned to the default project. And the first thing to change would be to assign those datasets here to our newly created project P1 and reorganize them. We can then save this to basically fix this change in our database. And then we come to the second part, and this is evenly important, and that is basically one of the whole purposes of building these types of data infrastructures, and that is actually linking the metadata to each of these FastQ datasets. So we have now here defined a set of columns to store categorical data basically that are assigned to each of these datasets. But that later basically drives the analysis. So for instance, we have the tissue type here, which is tumor or healthy in this case. We have the replicate information, and we have basically an alias here, which is basically an abbreviation of the data that we are storing in here. And we can just very easily add new columns here and change the structure of the metadata that we tag each of these data sets with. And the important part is that each of these columns is also represented in the database, and we can make use of these columns for our analysis later and describe our samples. And this is really an advantage doing this right away along with the raw data, because whatever type of analysis we want to implement later, we can always reference the metadata through the connection of the raw data. And also for the FastQ files, we wanted to have the option to actually upload files. So here in this case, we have now our project 1, and let's say for this first sample, we also want to upload some NGS stats, so basically some statistics maybe, or some summary on the next generation sequencing library preparation that we also want to collect systematically, more for documentation purposes, for instance. Then we can also simply upload this, and it will basically be attached to the FastQ files in our database. So once this is done, you can see that there are actually two more pages here. This is the RNA-Seq and the gene expression tabs. Those are more for actually analyzing the data. And before we will have a look at this in the second part of this video, we will dive into the bioinformatics that precedes those two steps. That was the first part of this case study where we looked at the data management and how to organize raw and process data from our research process. If you're interested, stay tuned for the second part where we will look at how to integrate an automated bioinformatics pipeline that is performing the RNA-seq data processing and also the differential gene expression, and how we can integrate the results of this also into our user interface. Stay tuned, have a look at the second video, and I see you later.